Thank you really for the invitation, and thanks, Asaf, for organizing this visit. It's really, I think I'm halfway through, and it's already very exciting. I meet a lot of people, a lot of interesting discussions. First time in Taiwan, so of course, first time in World Vegetable. Um, and today I would like to to give an overview of what we do in our lab in Volcani. And the title is Genetics and Epigenetics of Heat Stress Response and Resilience in Tomato. So I don't have to talk too much about climate change and how adverse are the effects on crop yields. So as other people might work on the more agricultural aspect and really taking care to build a surrounding an environment for the plants which be more suitable, we are coming from the plant's perspective, and we are asking what are the genetics and the genetic pathways and the genes that enable the plant to sustain better the stress. So, and more specifically, we are working in this field of gene discovery, and in the hope that by identifying the genetic and epigenetic elements that are involved in plant response to heat stress or any abiotic stress in general, we will be able to manipulate these factors in order to reach altered gene expression and trait improvement. So this is like the official title of the lab, Genetics and Epigenetics of Plant Abiotic Stress Response and Resilience. But really the main focus is heat stress response and tolerance in tomato, being a major vegetable crop and also a great scientific and genetic model. We have, as you know, a lot of resources. Many come from World Veg. And genetic tools, and of course, a high economic value. So talking about tomatoes, this is an example from a paper of modeling, so computational modeling, asking how tomato production will be in the next few decades. And what they did, they used several computational models to predict what will be the number of low yielding years per decade in the main areas, main location for processing tomato production, being USA, Italy, and China. Nowadays, China is the biggest producer of processing tomatoes, but still these are the three biggest producers. And you can see here that 2 out of the 3 models tested, that with the years in most places, the number of low yielding years per decade is dramatically increasing. And if you take it to crop production, the amount in tons, you see a clear and very sharp decrease. Again, 2 out of the 3 models. And why is that? So I'll give some brief introduction for the response to heat stress in tomato. Of course, tomato is a self-fertilization plant, and yield is dependent on successful fertilization, while pollen and ovules are in the same flower, staying with the same flower. So when you compare these two conditions, the normal or non-stress conditions with high temperature conditions. So this, for example, this is not a very acute stress, 36 degrees during the day and 26 during the night. But when it is prolonged and it is occurring for more than several days, then you start to see the effects mainly on the flowers, pollen, and fruit development. So here you can compare the bud, and you see the style elongation, which is much more profound when the flower opens. And this, of course, damages fertilization because the pollen cannot reach the stigma. These are healthy, nice-looking flowers, and here, this is really problematic, and it reduces yield. If you look inside the anther cone, what is going on with pollen grains? These are pollen grains staining, and the round shape ones, nicely stained with purple color, are the healthy, happy pollen grains under normal conditions. While when temperatures rise, you start to see a lot of abnormal pollen grains and misshaped and degenerated. Also, you can see that the tapetum, the tissue that is surrounding and protecting and nourishing pollen, starts to be degraded. So you see a lot of tapetum debris all around. And this is an electron microscopy image. You can see the round, very nicely ball-shaped pollen grains compared to the shrinked pollen under heat stress conditions, which will not function as well as these ones, meaning again low fertilization and lower yields. And this was demonstrated a long time ago by Marie Pitt in North Carolina. She was estimating the reproductive performance of tomato plants, comparing different average daily temperatures. She was comparing also fertile and sterile plants, but it doesn't matter because both show the same trend. So if you compare the average temperature of 25 degrees with 29 degrees, you see 89% decrease, for example, in the number of fruit per plant, and total fruit weight per plant is also dramatically reduced, as well as the number of seeds per fruit, again, because of the pollen sensitivity to heat stress. All in all, average fruit weight is reduced in about 40%. So if you take the effect on the fruit weight and number of fruit per plant, you can already see that the yield decrease is really, really dramatic. So I will, in this talk, I'll combine I'll tell you a little bit about several projects in the lab combining the genetic and epigenetic aspect. And this project, from the genetics perspective, aiming to find genes from wild species of tomato, which are normally more resilient to abiotic stresses, in order to incorporate them into the cultivated germplasm of tomato. And for that, we characterized a backcross inbred line population of Solanum pimpinellifolium. Why Solanum pimpinellifolium? So this is the lycopersicum clade, and Solanum pimpinellifolium is really is considered as the ancestor of the cultivated tomato. It is really close. You can really easily cross these two. It has red, although very small, but red fruit, not unlike some of the others here. And more importantly for us, it was developed in coastal regions of South America, in Ecuador desert. And this Pimpi LA1589 is the specific accession that was used to construct the population that we are working with. And it was developed in this region of high altitude and very high temperatures and low precipitation. And when we started the project, it was already published that this specific line is tolerant to salinity and water scarcity. But heat response was not tested by then. And what we had is a backcross population from Professor Dani Zamir at the Hebrew University, Faculty of Agriculture in Israel. What he did in his really impressive work, one of his very impressive projects, is to cross the cultivated tomato, the M82 cultivar, with this accession of Solanum pimpinellifolium. So the F1 generation was backcrossed to twice, and then selved for 9 generations to eventually result in 166 lines, each of them containing mostly the cultivated genome, but certain parts of the genome, each one has a different segment of the Solanum pimpinellifolium genome. And what they did is phenotyping for yield, performance in the field under normal conditions, no stress. And more importantly for us, they also produced genetic maps by SNP chip genotyping. So Danny was very generously giving us this population and the genetic maps. And then Neta Bashari, a master student in the lab, started characterizing, and we were interested really in characterizing the effect, how this population will respond to heat stress. So we started with asking, actually looking on all the lines, what is the coverage of the Solanum pimpinellifolium genome in these lines? And you see that it's quite variable, which is nice. We have different from like 0.038% to 16% coverage of the pimpinellifolium genome. And the introgression length also varies very much, from only 64 base pairs to 71 megabase, meaning we have a very big genetic diversity, very big. Well, it is all M82 and LA1589, but this is quite a considerable genetic diversity we can work with. And these are just few examples to say that we saw also high phenotypic variation in the greenhouse. Every time I entered the greenhouse, there was another trait that appeared to be varying with each stage of the plant. So if this is the pimpinellifolium fruits, which are small, you can see here that there are plants with round tomato, elongated tomato, and paternocarpic tomato producing no seeds. So we started by just evaluating the phenotypic variability to get a sense of how variable is this population. And this is still under normal control, non-stress conditions, just to say that we were able to detect variation in every aspect that we were looking at, either fruit-related traits, so the color, size, shoulder, peel, and fruit resin, meaning some of the lines presented fruit dehydration already on the vine, on the plant. Of course, flower-related traits, and also all other traits, which are, again, not so much directed into plant stress, into heat stress, but just showing variation. And these are the heat stress related traits that we were looking at. So when this population was grown under moderate chronic heat stress conditions, MCHS, meaning at least 20 degrees during the night and 32 degrees during the day, but for prolonged a period of time, what we observed, and these are few of the major phenotypes or phenomena that we find under heat stress conditions, we saw that this population segregates for anther deformation. So if this is a normal, healthy looking flower, under heat stress, you see many incidents of anther cone deformation. You see anther browning. So this feature is the result of Ross oxidation. So it can happen with many stresses, but it is getting worse under heat stress, under high temperatures. As we mentioned, elongated style, which is very much profound, and you can see a big variation in the severity of this phenotype between the lines of the population. And something that was very apparent is the lack of seed production, again, because of the sensitivity of pollen grains to high temperatures. Still, for example, here, few lines did manage to produce seeds. So, and this is just the distribution of the different traits under heat stress conditions. And what this suggested is that indeed we find substantial variation specifically for heat stress related traits. So we can further use this population in order to identify lines which are better tolerance to heat stress. And I will not go through the entire process with the entire population. But when we wanted, just to now elaborate a little bit of the next step that we took in order to screen specifically in a controlled manner and compare between controlled and heat stress conditions. This we had to do in a more confined environment, and space was more limiting. So we could not take the entire population of 166 lines, including parental lines and so on. So what we did is, because we had the genetic maps, we were able to construct a subpopulation, like a core collection, if you would like, that actually represents the entire Solanum pimpinellifolium genome. So if these are it's a summary, okay? It's just a summary of the genetic map for each of the lines that we selected, showing that the Solanum pimpinellifolium is found across the entire genome if you take into account all these lines. And these lines, these are only 224 lines. So these we can take and compare between the two conditions in a controlled environment. And this is the experimental setup. So we had two identical greenhouses with controlled, of course, controlled environments. So we could really maintain the same temperatures at the beginning. So the blue here, the blue color is for the control conditions, and the brown is for the heat stress, the MCHS conditions. So initially, the two rooms were exactly the same temperature. Upon flowering, we initiated heat stress in one of the rooms and maintain it as at least 22 during the day and 32 during the night, really to achieve the conditions of prolonged heat stress. And we validated it, we verified that indeed the plants sense heat stress by testing a molecular marker. So we are using HSP 17.6. This is a heat stress protein that is known to be highly expressed under heat stress. So this is, for example, the two parental lines, the M82 and the pimpinellifolium accession. Under control conditions, there was no transcript, no expression of this gene, whereas under the heat stress conditions, it was very much induced. So with this we could verify before we even got to the flowering stage. We could verify that indeed the plants are sensing heat stress, and these are the phenotypes that we were later on characterizing, because we are interested in the effect of heat stress mainly in the reproductive stage, to see how the plants will perform eventually. So to cut a very long story short, this experiment plus the screen of the entire population provided us with one line, like the first line that we've identified, with better performance under heat stress conditions. We denoted HTL1, and this line was showing better performance in terms of pollen characteristics. So, and this is a picture, just an example of pollen viability, Alexander staining, comparing viable in purple versus non-viable, and we also quantified it, but you can see by these pictures that M82 has about 50-50, like 50% of viable to non-viable pollen. The parental wild species, pimpinellifolium, has almost 100% of pollen viability, and the tolerant line is much more similar to the pimpinellifolium parent. We also quantified it. Nowadays, we are using flow cytometry to estimate pollen viability, and we use the DCF stain, which actually it's actually a reporter for the Ross quantity. And this was initiated in collaboration with Gadi Miller, our collaborator in Bar Ilan University. He was showing that under heat stress conditions, and this is Arabidopsis, so you really see a decrease in fluorescence, a decrease in viability. This was correlated with a decrease in pollen germination between the two populations of the two conditions. So what happens in flow cytometry, all the pollen grains are ran through this very, very thin nozzle, and a laser beam actually gets the fluorescence get the signal of the fluorescent stain. And eventually what you get is this plot where each dot represents one pollen grain. And you have the signal intensity, which correlates with grain viability. So on the x-axis, you have the actually viability measurement, and the y-axis is a measurement that correlates with size, because this is an estimation of how the laser beam is fragmented and is scattered. And this is affected also by size, among other things, but by the size of the pollen. So you can see that there is a subset of the population which is not viable. There is no signal. And there is a fraction which is viable. And again, there is a deviation. There are two subpopulations here. One, you would say like high viability or high ROS content versus the low ROS, which is also smaller. So we are now using this method in our lab, and we are looking at our lines. So this is already work done in our lab on this HTL line that we've identified in this screen. And these are basically the same plots, a bit different shape, but these are density plots. So the more like yellow and red is the more grains present there, and this is the quantification. And we can really mark the non-viable pollen by their level of fluorescence. And as we saw before, the viable pollen really divides into two subpopulations. If you want to, you can say like a smaller and bigger pollen. So what we see here is that under control conditions with this line, you see a very like a normal distribution of pollen, while under heat stress conditions, the viable pollen is really dramatically decreased to 32.68% of total pollen, which is a significant decrease, right? But when you compare to the sensitive line, the TA209, again, under control conditions, it's very similar. But this line, this sensitive line, under heat stress condition, totally loses viable pollen. I mean, this is reduced to like 8% or something like that. Meaning, this line, again, manages to maintain a higher fraction of viable pollen. Thus, also seed production is much improved. And we see a similar effect when we are looking at pollen germination. So these are, again, microscopic images of pollen germination assay. So you see here the pollen with the tube, with the pollen tube, and we count and quantify it, but you can already appreciate from the pictures that the cultivated line, the M82, is really having a hard time to germinate, while the wild species, the Pimpinellifolium and HTL1, are managing quite nicely. And if you quantify this, so the percentage of pollen germination, you see a substantial decrease for M82, while HTL1 is really similar with the Pimpinellifolium. We also quantify the length of the pollen tube. And again, you see a similar trend, M82 being very sensitive, while HTL1 is more similar to the Solanum Pimpinellifolium parent. Another thing we've identified just by accident, because we didn't really looked for it, but in one of our screens that was done in Gilat, the southern campus of Volcani, we noted that there is less occurrence, like a decreased occurrence of blossom end rot for this line HTL1. So we quantified that as well. And you can see that while most of the fruits were affected in M82, HTL1 was much less sensitive to this phenomena, as long as with other. So we are not carrying on with that, but that's just another piece of information, might be interesting. And again, going back to what I said before, that we actually have the genetic maps, but the genetic maps that were provided were very like initial, and they were the SNP chip data. When Yara joined the lab, even before her master's thesis, so I guess most of you know Yara already from her work here. So Yara started with her research project to actually to generate specific molecular markers which will enable the identification of each and every introgression, every segment of the Solanum pimpinellifolium genome. So what we have now, following her work, we have actually a coverage of all the genome with validated markers, and we are now going to test an F2 segregating population, first for linkage analysis and also for tail analysis using the DNA and RNA-seq. And what we would like to have is to really split these introgression and find linkage with the tolerant traits. So this is where we are at with the genetic project. Now, moving on to the epigenetic part, coming from my expertise really combined. I was also moving from genetic and epigenetic labs during PhD and postdoc. So when I started working on abiotic stress, so of course genetics is all around, but so 9 years ago, the effect of epigenetics was very much in doubt. So at the beginning, people were very even hesitant to discuss epigenetic as a source for breeding. But today, I'm much more comfortable discussing the concept of epibreeding, meaning really taking advantage of epigenetics for variation, for increasing variation, and use it for in breeding schemes. So just to compare the two sides of this playground, assessing variation, phenotypic variation, we have the genotype, the genetic code, which, as we all know, variation comes from a specific loci in the genome, and once we identify the locus responsible for the trait, we can integrate it into a breeding scheme. While the epigenetic segment is a more, I would say, elusive one, and it is highly and very tightly linked with environmental with the environment of the plant, which makes it actually more relevant in terms of fighting climate change effects. So one of the factors that that affects the environmental segment in this variation, phenotypic variation, is coming from epigenetic factors. And this is what we want to look at, asking what specific epigenetic factors would be involved in the response of heat stress in tomato, and what we can do to use them for increased tolerance. So then over the years, it became more and more apparent that many breeding important and agriculturally interesting traits are linked with phenotype, with epigenetic factors. And in a not very recent, but few years ago, we published a review focusing on epigenetic factors that are linked with very important traits in crop plants. So we totally excluded Arabidopsis papers, which is most of the publications were that. But still, there is quite a lot of an interesting and relevant and economically important traits which were linked to different epigenetic marks and traits. And I will not go into each and every one of these because we are mainly focusing on DNA methylation. So you can see that also DNA methylation itself was associated with many traits such as heat stress response, viral infection, reproduction, photoperiod, drought stress, and so on. So in parallel to this like recent proliferation of the linkage between traits and epigenetic factors and DNA methylation, we also gather a lot of data. So with years, we have more and more epigenomic studies, and we currently, and this is already quite old, but we already have DNA methylome maps of many important crops, including tomato, maize, rice, soybean, etc. So we have the phenotypic information, we have the epigenetic information, and we can really start to ask what are the causal relationships. So just briefly to explain what is DNA methylation, why is it important, what is it doing in the plant and the cellular function. So DNA methylation, when we say DNA methylation, we really talk about a CH3 molecule that it is covalently attached to a cytosine on the DNA, and it can come in plants in any sequence context. We can have CG, CHG, or CHH. I will show it later. And this type of modification of DNA can be found anywhere in the genome. And it is really affecting chromatin behavior and regulation, transposon silencing, RNA processing. And for us, we are focusing on methylation that is affecting gene expression. And this type of methylation is present either in the promoter of genes or gene body. So in gene promoter, it is, I don't want to say normally, but in some cases it was shown to affect gene transcription. So the more methylation you have, the lower transcription. It is not true for all genes, but in some genes it works like that. And for gene body methylation, it is still uncertain what is the exact role, but there are many assumptions. It might be involved in RNA processing or DNA editing, or some papers show that it might actually be relevant to fine tuning of transcription, so not on and or off, but a more subtle regulation. So more interestingly, DNA methylation components, like specific, and this is, again, this is in Arabidopsis, but still the components are very well conserved across the plant kingdom. And specific components of DNA methylation, the DNA methylation machinery, were shown to be related to heat-inducible genes. So more and more signs that this mechanism, this area of research will be relevant to heat stress response. But again, we are interested in tomato, not so much in Arabidopsis, although we work on that as well. And in tomato, what is very different from Arabidopsis, that is more sensitive to lack of DNA methylation. So where in Arabidopsis, the scientific community was able to produce all mutants in all enzymes and all proteins, and really compare and do great genetic studies, asking questions of how DNA methylation works. In tomato, it was much more difficult, and many labs struggled for years to get lines of mutants in DNA methylation. Luckily for us, a neighboring lab in Volcani of Professor Tzachi Arazi managed to produce a mutant, a tomato mutant, in the DDM1 gene. And this specific gene is a chromatin remodeler. So what the protein is doing is actually allowing the spacing out of chromatin, so the enzymes that put the methylation sign can enter and access the DNA and put methylation there. So when DDM1 is gone, you will have no methylation, not because the enzymes don't work, but because they cannot access the DNA. So this is the work of Tzachi, and he, in tomato there are two DDM1 genes, A and B, and he did, he CRISPRed each one of them individually and then crossed to get a double mutant. Okay, so here you can see tomato plants of the double mutant, single mutants, and the wild type, the M82 cultivar. And he said, okay, there is no phenotype for the single mutants. He was really interested in what goes on in the double mutant, and they measured him and his collaborator Nicolas Boucher measured the total amount of DNA methylation in these mutants, and they were showing that especially CG and CG methylation is dramatically reduced in the double mutant, not so much in the single mutants. But for us, coming to look at the response to heat stress, we immediately we saw that there is no point to work with that very miserable plant. And we went on testing these single mutants because, I mean, under the assumption that although general methylation might not be very well, very, dramatically affected, there might be specific differences in specific loci across the genome that might have an effect. And this, and of course, everything under stress, the plant rewires its genetic interactions. We might see something there. So Tsachi was also very generous in providing us the seeds and the methylome data of these mutants. And when we took when we grew these plants in our lab, so under normal growth conditions, still without stress, we could see a slight delay in development, meaning they had a little fewer number of leaves. Okay, but that was it. Then we went on to check the response to heat stress, and we started with the seedling survival assay, where we take 10 days old seedlings, we put them to 4 hours at 45 degrees, and then we let them recover for 10 days, and we measure the percentage of recovery, of seedling recovery. And there was the first surprise, because while under heat stress, the M82 was really, really collapsing and ended at 20% survival rate, both single mutants presented higher survival. So that was a bit surprising. And we went on to ask what happens on the reproductive stage, which is, again, more important and more interesting for us. So this is the same setup as I've shown you before, the controlled condition greenhouses. One stayed at controlled conditions, while the other we changed it to mono MCHS conditions upon flowering. And then when we tested the flower and fruit traits, what we saw is that there was not much of a difference on the number of flowers per inflorescence nor the number of fruit. But when we calculated the percentage of fruit set, meaning how many flowers per inflorescence were able to ripe and develop into a fruit, then we saw a statistically significant difference between DDM1B and the M82 background. And moreover, when we counted specifically the percentage of fruits that were able to set seed, specifically for DDM1B, we saw a really a much higher value compared to M82. So trying to identify the causal element for this tolerance, we carried out a transcriptional analysis of leaf. So we have all the comparisons, I mean, between the different lines and different conditions. And now we can ask which are the genes that might be responsible for this phenomenon. And now I'm leaving the transcriptional results aside because we are still on that. I mentioned that we got also the methylation maps. And what we saw there, we managed to identify a specific locus, specific site where we see decrease in DNA methylation. So here you have the different cytosines with the level of methylation for each cytosine. And with these are 4 independent plants of M82 compared with the 4 independent plants of DDM1B. And these ones totally lose methylation on this side. And interestingly enough, the methylation pattern really reminds the pattern of the double mutant, meaning CG and CHG. So CHG was not here, but the effect that we found was also CG and CHG. So now we are really trying to validate this result. And in order to functionally validate this, we are developing in the lab tools for epigenetic editing. So like we would alter the genetic sequence. Now we want to alter in specific place. We want to alter the DNA methylation and lack of time. So I will not go through all of this. I'll just show you the system that Tal was building in the lab. Master student, now PhD student. And we were using the CNR tomato mutant, which upon methylation of a specific location, actually what we get is ripening blockage. So the fruits are kept yellow. So what Tal did is construct this system with deactivated Cas9 fused to MSSS1, which is a bacterial methyltransferase. And upon directing this system to the CNR promoter, we get higher methylation of this segment. So now we can use this system to check if indeed our DMR in the DDM1B is affecting the response of tomato plants to heat stress. So just to summarize on the genetic perspective, we've shown that this build population of Solanum pimpinellifolium is very well adequate to study heat stress tolerance in tomato, and it shows really great variability. We identified 1, now 2 lines which show a better performance for different traits. So now our interest is to identify the causal QTL and hopefully genes. And on the epigenetic perspective, so I talked about the DDM1B mutant, which presents surprisingly better performance under heat stress conditions. We've identified a specific DMR, differentially methylated region, and we are now working on the functional validation of this site. So I'd just like to acknowledge group members, collaborators, now also Asaf. You will be added to this slide, and funding agencies. And thank you very much for listening.
Thank you so much, Michal, for this interesting aspect of genetic and epigenetic on tomato. The floor now is open for question. If somebody is interested to ask a question, here or online. We have any question online? No. Question in the audience? Yeah.
Thanks so much for the nice presentation. I'm happy to see your very nice pollen phenotyping results using impedance flow cytometry. Here at World Veg, we've been working with IFC for several years, and we still cannot get, in pepper, we can't get the nice separation that you get. I'm wondering if you have any recommendations or suggestions why we have these. Usually we get 2, we want 3 like you, but we can't hardly get them.
Okay. You mean the 2 that you get is the non-viable and viable?
I think so.
I tell you, I mean, the short answer is that this is not impedance flow cytometry. This is a sorting. This is the impedance is going on the electrical measurement, right? Electrical conductivity, and this is something different. This is I think that this is more sensitive because of the laser system. And the reason why we chose this technology over impedance is that I didn't mention that. Okay, I don't show this project here, but this system allows you to use different stains for different aspects of pollen, not just viability. So for example, we also use a stain for flavonoid content. So we are staining for flavonoids, and then we can see the different amounts of the different subpopulations of pollen with different amounts of flavonoids, for example, for another project. So it is more versatile. I can tell you on the other way around that I think that impedance flow cytometry might be more actually accurate in terms of correlation with pollen germination. This in that respect, impedance gets very, very good correlation. If you know Ivorio from Naimer. So yeah. If you are only maybe there is again, I'm not using impedance. Maybe there needs to be some technical work to see if you can get better separation. If not, you can compare with just laser flow cytometry.
Thank you. Some more question?
Okay, thank you.
No, I have more question. Yes, I have two questions, please. One to the HTL line. Did you notice some other trait that you can distinguish between susceptible and non-susceptible, or which kind of benefit those lines got to the other related to the heat stress?
The HTL specifically?
Yes.
So HTL1, I was showing the pollen characteristics, but actually in accordance with that, we also noted that it is way better in seed production. So it produces a lot of seeds in a way that if I go in the lab and I just take the bag with many seeds, it would be HTL1 from all the lines. And for other things, maybe Yara remembers, because did you with the BARD experiments, did you observe any other benefits of HTL1 compared to the others? So there we did not compare it with this population, but with other lines of the BARD consort. Like other traits where HTL1 was performing well under heat stress.
So
not related to pollen.
That specific project you are referencing, HCl1 was not compared to other PMP population. It was compared to other commercial and research lines that we tested for heat tolerance. And specifically HTL1, while not producing numerically like amount as much as other lines under control conditions, the reduction under heat conditions for seed-related traits and fruit set was very much like higher. Like
so
it was better in dealing with the heat. Like the reduction was lesser in HTL1 than other commercial
also
lines.
for fruit set.
Also for fruit set,
Yes, that I didn't know. Thanks
so much. Thank you, Yara. And the second question is related to the double mutant. The same question: did you notice how the other effects that the double mutant affect the plant, or you didn't produce seed, or you didn't produce fruit?
Mutant?
Yes.
DDM1?
Yes.
We didn't test the double mutant for stress because it is
just on seedling stage. For heat stress, what you done for this?
So we tested the single ones.
Yes. Okay.
Not the individual single mutants.
I understand.
Not the double, because
okay.
It was born stressed. I would know. Yeah.
Okay.
Thanks.
Some more question? No. So thank you very much.
Thank you.
I had a question.
Yes. Who is it?
It's Stepan here.
Yes,
online.
Please.
Thanks, Asaf. I'm Stepan, the director of the Social Science and Nutrition Program, and I thought that your research is really relevant for World Vegetable Center, and I was happy to see that Asaf will be mentioned in the future on your acknowledgement slide. So I just wonder, like, what kind of collaboration are you planning with World Vegetable Center? Thank you.
Asaf, can I answer that? Yes. Okay. No, I'm happy to, So I'm really happy to say that with Asaf, and now Yara joined to actually do the work, We are mainly interested in the differences between wet heat stress and dry heat stress, and comparing the same germplasm, specifically the supermagic population that Asaf developed here, and compare the performance here and in my lab, my greenhouses, which actually ultimately we can achieve the same temperatures, but the humidity is way different, and try to first to ask, are there any common genetic interactions or common lines? I mean, would the lines that are better here would be better there, or vice versa? So that's the main goal of this project. Maybe Asaf can also
And the idea that we
elaborate.
mentioned today, that also that we can take it also regarding to the genotyping to
But that was on the
Yes.
on the biotic stress.
on the biotic stress, but also to measure some kind of genetic approach on biotic stress to see if we can identify some kind of overlapping or some kind of how the effect of BW6 on the affecting on the TY gene on the genome, on those ones that we cannot identify the TY marker on chromosome 6.
Yeah. We just gotten just walking around the field, we got an idea of how we can maybe improve the markers. And actually talking with people here, we already have some more ideas for collaboration. So that's very interesting visit.
Productive.
Very productive already. Yeah.
Yes.
Thanks.
It answer your question, Pepen?
Yeah, thank you so much. I wish you good luck. Yeah, thanks.
Thank you.
Thank you so much, Pepen. Thank you, Michal. And thank you, everybody, for joining us. And